Welcome to the University of the Free State.
Wa amohelwa ho University of the Free State.
Welkom by die Universiteit van die Vrystaat.
Siyakwamukela eNyuvesi yase-Free State.
Situated in the heart of South Africa, my university is a creation of the past, the present, and the future. Across our 3 campuses, 7 faculties have been home to some of the most magnificent minds. Everything we do is rooted in our values of excellence, innovation and impact, accountability, care, social justice, and sustainability. We have been changing the world for more than a century. We continue to pave the way and inspire a fresh stream of knowledge and skill to follow in the footsteps of our scholars and alumni. We hunger for knowledge that will help us to be better and to do better. Our facilities encourage excellence, pulling us towards the light of new insight and ways of applying what we know. We lead by example, advocating sustainability in everything that we do. While we are making a current impact, we are also steering towards the future with the knowledge and skills we need to take the lead. The next years will be especially significant in our growth as our Vision 130 unfolds, which will peak in 2034 when the University of the Free State celebrates 130 years of existence. Vision 130 is our strategic intent to reposition the university for 2034. Join us on this journey. Together, we will go further.
Please be seated. Good afternoon. Good afternoon. Yes, only Lisa would say that. Dear friends, colleagues, distinguished guests, ladies and gentlemen, good evening. An inaugural lecture is more than an auspicious occasion. In fact, if you can move it a little bit to this side so that when Professor Botma Visser is much taller than me. Thank you. Inaugural lecture, ladies and gentlemen, is more than an auspicious occasion, not just for the university, but for many of us, and in particular for the person who will be delivering it. For us, it allows us to acknowledge the appointment or the promotion of one of our own colleagues to the title of full professor and introduce them not only to the academy, but also to the broader community within and beyond our university. Ladies and gentlemen, tonight we have the privilege and the honor of introducing you to Professor Botma Visser. It really is a personal and a singular privilege that he has given me to be able to stand in front of you. In our midst, in addition to those that are in the audience, we have members of the university executive, senior management, our acting dean of the Faculty of Natural and Agricultural Sciences, Professor Johan van Niekerk, who has just ran the marathon. In the audience, we also have the other Vice Dean, Professor Liesel Hasselmann, and many others who are equally distinguished in our midst. Our Head of Department of Plant Sciences, Professor Adre Minang Ontong, colleagues, and I am so happy and delighted to see so many postgraduate students this evening. When I was a postgraduate student a few years ago, I think it was in the 80s, I was instructed to read about a very distinguished gentleman called Jan van der Planck. If you're a postgraduate student in plant sciences or in plant pathology and you have never heard of Jan van der Planck, you need to come and talk to Professor Visser so that he can take away your degree. He is one of South Africa's most distinguished early pioneering scientists who many of us learned about, and every generation develops their own Jan van der Planks. Every generation brings people who we look up to. And so tonight it really is my singular honor and privilege to welcome you not just to this occasion, but to the inaugural lecture of Mr. Rust, Professor Botma Visser, on his inaugural lecture, Battling Wheat Rusts in the Southern Africa, my part of the story, because that story has many parts. There are many of us among us in this audience who play a part in the continuous battle between the plant and the pathogen. Before we begin, allow me to express, Botma, my sincere appreciation to you to allow me to play this role this evening. It is an important event in your timeline. It is particularly important that it is the year that you deliver your inaugural lecture. It is also the year that you joined the 6th floor. If you don't understand me, he turned 60. It is an opportunity to congratulate you and publicly wish you the very best, because this is just but the beginning in more ways than one. Tonight is about Professor Visser, who has in excess of 36 years and counting of experience in plant pathology. You will hear a lot more about him from our head of department and our acting dean. But he got his undergraduate cum laude in the 80s. His master's and PhD in plant molecular genetics, 2004, all of them from this very institution. He's dedicated his career to studying the molecular genetics of cereal rust pathogens, particularly the rusts of wheat, maize, oats, sunflowers, among other plants, highlighting the evolving challenges these pathogens pose to food security in Southern Africa and beyond. We owe him a lot, and we owe others a debt of gratitude every time we have a slice of bread. This work was initially done in collaboration with another distinguished gentleman who this university has produced over the years, Professor Saki Pretorius, whom, again, if you don't know who he is and you're an undergraduate or a postgraduate student, you need to come and talk to Professor Visser. His work addresses many areas: genetic analysis, molecular characterization, and investigation of the presence of virulence and fungicide intensivity in wheat in South Africa. He is a scholar of note. I don't even want to even fathom to think how many peer-reviewed publications that he has produced over the years. But if you're a plant pathologist and you don't publish in plant disease, in plant molecular microbe interactions and phytopathology, and in molecular plant pathology and other journals of note, then you haven't yet started. He has co-authored and also mentored many postgraduate students. Colleagues, tonight, Prof, it's your night. It is for us to come and recognize and applaud your achievement, the leadership that you have continued to give to us. We take pride and pleasure in celebrating this significant achievement, and thank you for giving us your time, for choosing the University of the Free State as your home, and to inspiring particularly a lot of young people who I hope will follow in your footsteps. In keeping with tradition, I am now going to request the Acting Dean of the Faculty of Natural and Agricultural Sciences, Professor Johan van Niekerk, to formally introduce Professor Visser. Johan.
Thank you, Dr. Malapu. Good evening, colleagues, students, friends, and distinguished guests. It's a privilege for me to introduce tonight Prof. Botma Visser, a scholar whose journey has been intertwined with the University of the Free State for nearly 4 decades. That journey began in 1986, when he first walked onto this campus as a botany and microbiology student. From those early days, he has grown with the university, completing his BC Honours Cum Laude, his MSc and his PhD, and rising through every single academic rank, from laboratory assistant and junior lecturer to full professor, we celebrate today. Professor's passion for molecular genetics began in his undergraduate years, when a lecture on DNA changed the course of his career. Guided by mentors like the late Prof. Chris Small and Prof. Obas Pretorius, he developed a lifelong commitment to understanding the most elegant tools in biology, genetics, and bioinformatics. Over the years, he has built a remarkable research profile, particularly in the field of wheat rust. His collaboration with colleagues at the UFS, the ARC, and internationally has produced more than 40 published manuscripts, and has taken him to conferences around the world. He supervised and mentored a large cohort of postgraduate students, many of whom now hold international roles in academia, industry, and research, extending his impact far beyond his own laboratory. An important recognition of his scientific standing is his C1 rating from the National Research Foundation. And I want to say it again, a C1 rating from the National Research Foundation, a huge achievement, Professor. This rating is reserved for established researchers with an international reputation and for the quality and impact of their work. It is a clear acknowledgement that Professor is not only making a difference locally, but his contribution to molecular genetics and plant pathology are recognized at a global level. Beyond his research, Professor has always remained deeply committed to teaching. For more than 40 years, he has been inspiring undergraduate and honor students in molecular genetics, while also serving the department as a leader and a mentor. Yet, if you ask him where he is the happiest, it is clear. You'll find him in the lab, with his students, in the greenhouse, or out in the field. And Professor, I've spoken tonight to some of your students who are sitting here, and they say you have a very, very, very special way of talking to students in labs and in classes. And those ones who are shy, you have a way to get them to participate, and they really appreciate that. This evening, we celebrate not only an academic of great achievement, but also a teacher, a mentor, and a colleague who has dedicated his career to advancement of science and to the growth of our university. Ladies and gentlemen, please join me in welcoming Prof. Botma Visser.
Thank you very much, Prof. Manapu and Prof. Johan, for introduction that sometimes I did not recognize myself. It's right, now it's past. It is a place that I, at one stage, really did not think that I would reach. But to be standing here is an amazing privilege. So I hope that through my talk tonight, you share the passion that I have for what I do. Because I think I have the most perfect, most enjoyable, fantastic job in the world. And I enjoy it very much. So last year I attended a postgraduate symposium in Johannesburg, at the University of Johannesburg. And I actually, without knowing at that stage that I was promoted, already started to prepare the talk. So it comes a long way. And it is really a story. So I will try to relate the story to you. And I hope that you can keep understanding what I do. But, yeah. So you'll see in the title, I mention a battle. And the is depicted on this whole picture on this side. Let's start at the left-hand side. You are a farmer, you have planted wheat in your fields. One morning you stand up, coffee on the right-hand side, and then you see that your wheat fields look different. So you get into your bakkie, and when you get to the field, you see some of these plants are really very much diseased. So being a farmer, you pick a leaf, and at the back of the bakkie, you take out your research lab, and under the microscope, you look at it, and you see this. And being a very rich farmer, you take out your electron microscope, and you see this. And then you realize you have a problem. So you are now in the trenches. So what do? You phone a fungicide distributor, a gifsmoes. And he comes on the farm, and he provides you with a fungicide, and you spray. But usually when your plants look like this, it's too late. So then you do the next best thing, and you phone Dr. Terefre from the ARC Small Grain. He's sitting in the audience. Can't see now where he is. Tarakeng, there at the back. And he flies down to your farm, and he picks a few leaves, and he tells you this is a rust. But we need to identify which variant it is. So he goes back to Bethlehem, and he then sends some of the leaves to my two colleagues, Prof. Sakkie Pretorius, and then Prof. Willem Boshoff. And Prof. Mlapu, they are actually the rust guys. I'm just a tag along. And together, these three are the reason that you sit here tonight. And I want to really acknowledge them. Because if it was not for the work that they did, I would not be here. So Willem, and then I want to include Cornell as well, and Tarakeng, and I know Prof. Sakkie is online. So thank you. It is on your broad shoulders that I stand tonight. So together, these three tells the farmer, this is the variant that you have. You need to make a plan. When you plant again, you can start with a systematic spraying all through the growth season of the wheat. Or you can contact the plant breeders, Doc Adria, and ask, is there a new cultivar with resistance? And this whole scheme worked for many years, whereby they battled the rusts. And then in 2007, Prof. Sakkie asked me to join them. And I would like to think of myself as a profiler, where I do genetic analysis based on the work that they did. And this is the whole battle. But the battle is not being done alone all my students, and I got quite emotional when I saw all of them, MSc students and my PhD students. That plays a major role in what we do. So if we are the good guys, who's the bad guys? The rusts. There are three. On the left, Puccinia triticina, wheat leaf rust, Puccinia striiformis, tritici, stripe rust, and graminis tritici, stem rust. So these are the symptoms, disease symptoms that you can see on the leaf. If you look a little bit closer with your microscope, you see lesions like this that we call uredinia. So that is typical for leaf rust, typical for stripe rust, and typical for stem rust. And if you go to your microscope, these are the urediniospores. So these are the little spores that are released from the uredinia. Now, luckily for us, this battle has one of two possibilities. Luckily, I see that there's 4 bottles of water here. So please excuse me. In the rusts, there is a gene-for-gene interaction. What we mean is that in the pathogen, there is a dominant gene, which we call the avirulence gene. In the plant, there's a dominant gene known as the resistance gene. So when the plant is infected by a pathogen and both of them have these dominant genes present, the plant survives and it is resistant. And why? Because these two proteins that are encoded by these two genes bind to each other. And once they bind, then we don't have disease at all. The plants survive. But the pathogen is a little bit sneaky. It can change. It can mutate. So instead of a dominant AVR or avirulence gene, now it changed. It mutated. And eventually, the structure of the protein is now different. And then there's no binding between the resistance protein and the avirulence protein. And once that happens, you end up with disease. So such a variant we call a different race than the previous one. And that is the job of the pathologists. They would get the samples from the field, and by analyzing them, they can identify the different races of the pathogen. Once they are finished, they give me leaves with little lesions on, and then I start to play. So just a little bit of background. The genetic analysis that I do initially work on microsatellite markers. So microsatellite markers are also what they use in CSI and all those nice stories. It is DNA sequences that are repetitive, CA, 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 CA. So you have those that are 2 nucleotides, 3, 4, 5, 6, and so on. And the lengths of these differ. But for me and my children, for instance, those sizes are more or less the same because we are related. But for me and anyone in the audience, Habre, it will be different. And based on that, we can determine the genetic relationships between the fragments that we PCR amplify over here. So we use that data. You'll see a lot of these tonight. This is a phylogenetic tree. Those in blue, those 4 are closely genetically related to each other, while they are further related, much further away from those in green. And we can use that same data to do structure, where all the related isolates are divided into a certain subpopulation that differs from another one. Okay, so that is the past. Now the work that I started. In 2007, Prof. Saki pulled me in, and this was done about in 2015. We got a number of herbarium specimens for Puccinia triticina leaf rust. They were collected between 1906 and 1980, different places. Remember this one. It was collected in Tanzania, Tanganyika, in 1945. So when we compared these herbarium specimens, those in bold, with our current isolates or races in the normal script, you can see some of these subclades consisted only of current isolates, some only of herbarium isolates, and some is a mixture. So what does that tell us? It tells us we can divide them in sublineages based on their similarity, 5 of them. And then we can get a timeline. So what we saw was that there were 5 of these genetic lineages. The one longest in the field started in 1906 and it lasted until 1988. Very long time. And that is only based on our current isolates and those herbarium specimens that were available. Then we had another one that started in 1909 to 1948, and another one from 1926 to 1988. These 3 are extinct in the field, so they don't matter anymore, but at least we know that they were present there. Then we have 2 current lineages, one from 1984 to 2005, and this one from 1985 to 2010. But remember the 1945 isolate? Tanzania, yet it shows similarity to our races that we have. Remember that. I'll get back to that. So as I said, Tarakeng and Willem, Prof. Saki, they would every year, or actually Tarakeng does that, he goes on annual surveys, collect isolates from the farmers' fields and so on, characterize them, and then they discover new races like 3SA170, 3SA100, and 3SA127. And what I then do is do the genetic comparison, and you can see 170 grouped with 3 of the older isolates or races. These 2 with a 3SA146, also an older one. And if we then put it in structure, now we can see this is lineage 4, but lineage 1, it can now be divided into 3 sublineages. Hope you're still with me. And we can adjust the timeline. So instead of having a single lineage 1A, we now have 3 subgenetic lineages. And that tells a lot, because now we know that this population is changing. And we know how it is changing, because they have done the virulence analysis. They know which virulence genes was added to the pathogens. So that was leaf rust. I actually wanted to do the other 2 rust also in detail, but that would have been a little bit boring. So I'm just going to summarize Puccinia graminis, stem rust. Again, we have a number of old isolates, Zambia, Uganda, Mozambique, and again Tanganyika, together with some of our South African isolates. And here we see there was just 4 genetic lineages initially, 2 of them up to 1945, old lineages, and then 2 new ones, 3 and 4, and they were represented by initial races that were first described in South Africa. We'll get back to that. This gap does not mean that there were no stem rust at that time. They were just not collected in the herbarium. So what are our conclusions? This population is dynamic. It changes all the time, and we need to keep up. So the work that Tarakeng does is essential. Every year we have to do the surveillance to make sure that we know what is new in the field. And then our population is structured or shaped by new mutations within our current isolates in South Africa, but also due to exotic introductions. So what is an exotic introduction? And maybe down under is a nice place to go. Down under is Australia. We know it's an island in the middle of the Indian Ocean. Yet, in 1920-something, they received an unknown isolate. This date is a little bit wrong. In 1954, another one, and in 1962, 2 others. And they formed the basis of new genetic lineages that developed in South Africa. In Australia, apologies. So now you can ask, where did these 4 isolates come from? What about out of Africa? Because at that stage, there were similar isolates in southern Africa compared to Australia. Now, those isolates in southern Africa were not kept, they were lost. So for this study, we used our current isolates that we have, or current races. So the hypothesis was that these isolates moved from southern Africa across the Indian Ocean into Australia. If we need to prove that, we need to do 2 things. The first one is to show that our current isolates show some genetic similarity with the Australian isolates. And that is exactly what I did as the profiler. We saw that one of our genetic lineages, the UG99, was totally different from everything else. Their race 1 to 6, totally different from anything else. But here we have a mixture of South African and Australian isolates, showing that they are genetically related to each other. We can see it over here, UG99 unique, race 1 to 6 unique, and then here we have a mixture of South African and Australian isolates. This is maybe just a little bit a better indication. This is UG99, way different than race 1 to 6, but these are a mixture of our races and theirs. Now the difficult part came, that I did not do. I want to say it beforehand, because what this guy did is amazing. When we go on a conference, they take us through their fields where all their trial plots are planted. And many times, when you walk through it, on your shoes, you get these urediniospores. So the next day you climb on the plane, you fly back, and these spores travel with you to a new country. But the problem was, in 1920-something, travel between South Africa and Australia was very little, because not a lot of flights moved between the 2 countries. More importantly, that was also true in 1954. So how did it get there? It went with the wind, as a hypothesis. But can we show that? We know that in the southern hemisphere, this is Antarctica, there are westerly winds blowing from South America, across South Africa, across Australia, and back. So there's a means of transport of these spores. I would advise people that have hay fever, take out the tissues. This is a field in Spain where they are harvesting triticale. And that triticale was infected with stripe rust. And everything that you see, that cloud, are urediniospores. So you can imagine it's a warm day. There are thermals going up. So as you cut this triticale, these spores are released and they travel up 10,000 meters where the Boeings fly. So this was the clever guy, Marcel Meyer. He was from the University of Cambridge. He actually sat next to me at a conference and asked me what I did, and I asked him what he did, and eventually we collaborated with each other. So he's a computer guy. So he did computer simulations where he took characteristics of urediniospores. Remember now, that is these small structures. He used 14 years of, I always struggle with this word, meteorological data of Southern Africa, the Southern Indian Ocean, and Australia. And then he used survival time of the spores of 3 and 5 days, and he simulated it from 5 sub-Saharan localities and to determine whether these spores can actually move. So this simulation is from Bethlehem in the Free State. Where it is dark red is where there's a high concentration of spores, and these yellow wisps, very low amount of spores. But hopefully you can see every now and then that spore cloud just touches on Australia. This was when he said for the simulation that these spores can survive 3 days in the air. When he increased the survival time to 5 days, look at the difference. Now those spores goes right over Australia. And that led us to believe that movement of spores from South Africa to Australia is indeed possible, as they have hypothesized many years ago. So this is the simulation. You release urediniospores in Bethlehem, and then sometime it can move over Australia. Interestingly, when you use the Western Cape as the place where the spores are released, they don't even make it halfway over the Indian Ocean. So, but what about South Africa? I've showed you that some of our genetic lineages starts at a given time. We get our introductions from North Africa or Middle Africa, Mid-Africa. So here we can see is chose Tanzania and Zambia and Zimbabwe as release points. And now we have the spore cloud from Tanzania cover Zambia, the one from Zambia covering basically the whole of Southern Africa, same with Zimbabwe. So we get our foreign introductions from Mid-Africa. So we need to go back to know what we must do in the future. So what do we need to know? Wind plays a major role in shaping our populations of the rusts. It is important that we know that. It is important that we know what happens in South Africa because the Australians depends on us knowing what is happening in our own country. Let's say that they get a new introduction. And we know that we receive our urediniospores from Mid-Africa. Okay. So that is the conclusion of this study. As a profiler, I wanted to know more about these rusts. The best way to control them, that I told you in the first slide, is to spray them with a fungicide. But pathogens can adjust. They can mutate to become insensitive to these fungicides. So the slides are not very clear. This was Isabella's master's degree. But we showed that even at a 66-fold dilution of the fungicide from their working concentration, we still had total inhibition of germination. So we came to the conclusion that there is no insensitivity against fungicides currently in South Africa. So Isabella also sequenced the target gene CYP51, and we got a single mutation where we have an amino acid change, but it did nothing to the functionality of this protein. So we are, in terms of stem rust, very safe in terms of fungicide insensitivity developing. Then a study by Emily, who's also here tonight. She looked at some of the avirulence genes, AVR-SR35 and AVR-SR50. You can see when infected, a cultivar carrying the resistance gene was resistant against infection. And when she sequenced the alleles, all of them fell within a clade that represents avirulence alleles, telling us there is no virulence against SR35 and SR50 in South Africa. So what did this teach us? It taught us no fungicide insensitivity for stem rust, no virulence for these two resistance genes, but they took quite a while to complete the study because it's labor intensive, it's time intensive. And this is the final part of the talk. Is there an alternative approach? So the approach was, we have the phenotype, and then we do the genotype. So you go from what the plant looks like to understanding what the gene does. Can we change it around, that we first look at the genes, and from that we determine the phenotype? And this is a program that we started with Isabella's PhD, and it is called MARPLE, and it was developed by a lady, a collaborator of us in Norwich in UK, Dr. Diane Saunders. It stands for Mobile and Real-Time Plant Disease Diagnostics. So the idea is that you can travel with a bakkie, with a generator on the back of the bakkie, and then do everything that is indicated over here. So you start off by having a lesion on a plant, extract DNA, and then you do a PCR amplification. Actually, 4 of them, where you amplify 400 highly variable genes. So these genes will allow us to do the genotyping. But included in this group of genes are also those fungicide sensitivity genes, or sensitive genes, as well as avirulence genes. Then we purify the PCR reactions, we barcode them, and then we run it on this little marvel known as a min-ion personal sequencer, and we can sequence 8 isolates at a time. That data is then, through a bioinformatics pipeline, allows us to do 3 things. The first thing is, it gives us an idea of the relationships between the new isolates and our local races, as well as global isolates, because that is important. What happens in South Africa, we cannot leave here. We have to link that with international work. And then we can look at fungicide insensitivity and also the presence of possible virulence alleles. So we did the trial right at the beginning, 2 trees. You can see the topology is very similar. This one is a little bit better. That was generated for the same isolates using microsatellite markers. We used 20 loci. And then this was done with MARPLE, where our number of loci increased to over 400,000. So the statistics on this one is much better than on that one. I gave a talk yesterday in Pretoria, and I had to stop you and ask, is this not beautiful? Huh? Isabella, this is really very nice. This is a study, part of a PhD, and it is on stem rust of wheat. So in this part, clade, and over there are our South African isolates. So she worked with 96 such isolates. These that are not colored are international isolates. So they were already characterized previously. And if I zoom in, you can see all our non-UG99 isolates grouped with a number of isolates coming from South Africa, Kenya, and Australia. Remember the movement of the spores to Australia? So this shows that Australian isolates, again, are similar to ours because it moved from us to Australia. If you go over there, that is the UG99 group, and they grouped together with a number of isolates from mid-Africa, Kenya, and I can't read there, Uganda, and so on. Again, confirming the movement from mid-Africa to South Africa. This works, Isabella's still busy, but I think this is the main thing that we are looking for. So from that sequence data, she looked at 2 specific genes, succinate dehydrogenase and this 14-alpha-D methylase. This one is targeted by the STH class of fungicides, this one by the DMIs. If we look at this one first, there are a total of 12 mutations in all our isolates. These 8 or 10 at the bottom, you can see they are present in literally every allele. So I am not too worried about these mutations because they are present always, and we have previously shown that all our isolates are susceptible to the fungicide. But these 2 are a little bit of a worry, 38 and 377. So the problem with these are that the mutations are very early in the gene. If it is a nonsense mutation, then we have a stop codon, and then the rest of the protein will not be produced. If it is a missense mutation, that might not be a problem, but this is the next step for Isabella. Over here is the DMI fungicide, CYP51 gene, 8 SNPs, 4 alleles, and that also need to be analyzed in future. Okay. Second last slide. What has happened over the last 17 years is that I started just looking at microsatellites, and this evolved whereby now can look through MARPLE not only at fungicides, at the genotyping, but also at different genes that might play a role in the survival of the fungus. So we went from phenotype to the gene or the genotype initially, but this changed now from the genotype to the phenotype. So with this single approach, we can do 3 things. And for the South African wheat production, it is important that not only do we succeed in this, but that we expand that. And one possibility to expand is maybe a Southern African collaborative project. So we have collaborators in Zimbabwe. Isabella knows someone in Zambia and someone in Kenya that works on the rust. So the idea is maybe they can send us samples that we can extract DNA and combine that in this big MARPLE analysis. And now they have a very weird isolate, totally different. In some time we will receive that one, but we can be prepared if we know that it is on its way. And that is what we hope to do in future with this work. So Prof. Saki, in the early 80s, started with this rust program. I think it's important that his legacy is taken forward. That after retirement, this program still runs, because we have invaluable isolates and data that I feel must go forward. Right, so as Johan and Prof said, next year is my 40th year at this university. 4 of them was as an undergraduate student. 36 will be as a lecturer. And as I said in the beginning, I've never had a bad day. Now I'm lying. I have a few bad days maybe at university in the job that I do, but I thoroughly enjoy my work. But I know 10 years after my retirement, no one will remember my name on campus anymore. After I published my last paper, 10 years, I will not be cited anymore. Where am I going with this? The writer of Ecclesiastics in the Bible starts off by saying, everything is worthless. Vanity, he calls it in English. Meaning, you put so much effort in what you are doing, is it worthwhile? And then he ends at the end of the book, and then you die. A little bit morbid. But in the middle of the book, he writes this beautiful passage. Go, eat your bread with pleasure, and drink your wine with a cheerful heart, for God has already accepted your works. Whatever your hands find to do, do it with all your strength. And I hope that's where I am at this stage. I have 5 years until retirement, but there's so much more that I want to do. And I enjoy what I'm doing. So I'm not a very philosophical guy. But if I can end up with saying this, to my own children, Colette, Carita, and Henlu, to my lab children, Isabella, Amy, Yaki, and Emily, and to my third-year children, Eni, Gofatsu, and Mamelu, yeah, I'm correct. I've wrote this down. May your passion turn into your purpose, and may your purpose turn into your pleasure, because that's how I feel about the work that I do. So thank you very much.
And so the battle continues. It is such an honor for me to stand here both as a former student and now a colleague to celebrate this remarkable academic and personal milestone with Prof. Botma Visser. From everything that you have heard tonight, it is clear that Professor Visser is more than just a teacher or a researcher. He doesn't merely convey or transfer knowledge. He molds, he mentors, and he inspires. He shapes the next generation of wheat rust researchers with purpose and vision, making sure that the pursuit for food secure future continues beyond the boundaries of the laboratory. His work has real-world impact, helping to ensure that millions of households across Southern Africa have access to one of life's simplest and most essential foods, bread. I enjoy my bread thoroughly. Wheat rust remain among the most persistent threats to global wheat production in Southern Africa, where wheat is both a staple crop and a pillar of food security. The challenge is especially urgent. However, Professor, with his unwavering commitment to wheat rust research and his scientific leadership, has placed South Africa at the forefront of plant disease surveillance and response. His work stands as a beacon of scientific excellence, blending rigorous research with practical solutions, offering not just data, but hope and resilience to sustainable agriculture. Prof. Botma Visser, once again, congratulations on a magnificent, insightful inaugural lecture and on a career that continues to inspire, one that ignites young minds and encourages seasoned ones to literally, quite literally, unrustify. Then, I just want to do a word of thanks on behalf of Professor Botma. He's thanking everybody attending, especially those that traveled from far, and the ones that's attending online. He extends his gratitude to his family and his support system, his colleagues, friends, and students from the department as well as from industry. Then he wants to thank Dr. Malapu Kobela for the opening and the welcoming, Professor Johan van Niekerk for introducing Professor Visser, then of course Alicia and Dr. Nita and the team for organizing the event. Then he also wants to thank Sonique Productions for the sound, Roystool TV for the online streaming services, DigiPix for the photographs, and We Smiled. How does your face feel, Professor? Then, of course, the Natural and Agricultural Student Council for their assistance tonight. And last but not least, Dr. Cornell Bender for her invaluable support during this preparation of the event. Thank you so much. And and then Professor Visser would like to invite you for some refreshments in the foyer. If you will just allow him a few, would it take seconds or minutes? I don't know, with 60, to get to the door for congratulations. So if you can just remain seated until he reaches the door. Thank you.